Hi, everyone. My name is Michael Stewart. I am the Director of Education here at the OI Foundation, and we want to welcome you to today's session, which is the current state of OI research, an update for the OI community. Today's session will be an hour-long conversation where we're going to be doing a broad overview of what's been done in OI research over the past few years. We ask that you please keep your microphone muted throughout today's entire session. Thank you so much for that. And also, we're going to be covering lots of information. We're going to be directing you to different points throughout the day to the chat line. Also, with that being said, I want to introduce Tracy Hart, the CEO of the OI Foundation. Tracy.
Great. Thanks, Michael. Thanks so much for everyone being here this evening or this morning, wherever, this afternoon. I'm looking at Jeffrey Esslinger in Hawaii. So glad everyone is here tonight. I have the great pleasure of kicking off the meeting by introducing our Board of Directors President, Cameron Penn, who is going to be leading us tonight in this interview style session. We're really excited about that because we want to hear your questions as well. So Cameron, I'm going to turn it over to you so you can introduce Dr. Lee.
Thank you, Tracy. Before I get to Dr. Lee, as Michael mentioned, I do want to stress, if you're not familiar with the chat button right down there at the bottom, we will be taking questions later on, and we will take as many as we can possibly get to. So if you do have questions, post them on the chat. We will choose as we go. If there are questions that are similar, we will try and condense them and combine them as much as possible, but we will try and get to as many topics as we possibly can with Dr. Lee. So without further ado, I do want to introduce Dr. Brendan Lee. He is the professor and chairman at the Department of Molecular and Human Genetics at Baylor College of Medicine in Houston, fellow Texan. So Dr. Lee, welcome. Great to have you with us as always.
Great. Thank you, Cameron. It's a pleasure for me to be able to participate in this first of a series of conversations about research.
Excellent. We have a ton of stuff to get to, and one of the reasons we are doing this, and one of the reasons you've seen us on a lot of these virtual events, is over the couple of years ago, we made a conscious effort to be more transparent in how we share what we find through research, studies, surveys, among other things, and this is just another step in that process. So I think the first thing is especially with OI, is we have a lot of acronyms that are out there, and BBDC is one of the most prominent ones. Brittle Bones Disorder Consortium. What is the BBDC? Why is it so important that the OIF be involved with it?
Great. Do you see my first slide?
Yes.
Wonderful. So, part of the reason why I'm here is to really begin to talk directly with the OI community about some of the work that the BBDC has done. This has been a great partnership among many, many individual scientists, the OIF, the families out there, and I think we should be all very proud of it. So what you see in front of you is a map of the Brittle Bones Disorders Consortium. And simply put, this is a collaboration among many, many stakeholders. I've mentioned some of them, but driven by outstanding clinical and research centers around the country. You see the map in covering many of the sites in North America. And it's a multi-center study that's funded by the National Institutes of Health. And the goal is to perform clinical research, but broadly defined. And we'll get into that, I'm sure, in the conversation. But importantly, the Osteogenesis Imperfecta Foundation, as a representative of the whole OI community and even beyond the rare bone disease community, is a full partner in this research. And one of the really major, I think, reasons why the OIF has been so involved and has been such an amazing partner is that it really drives the momentum of the field forward. What we have seen as the BBDC is now one of over 20 rare disease clinical research consortia that's funded by the NIH as part of the RDCRN, the Rare Disease Clinical Research Network. We are the only one focused on bone and bone health, and specifically brittle bone diseases, of course. Together with the OIF, I think we, over the past 7 years, have built this enormous momentum that I think has the potential to really transform how we take care of patients, how we bring new drugs and treatments to patients, and how we really educate healthcare providers and the next generation of scientists. For all those reasons, I think the OIF's commitment and support and partnership has been so critical.
All right, so let's get down to the nitty gritty. What kind of research are you doing that's different from other studies that are out there?
Absolutely. So first, let me say a little bit about research that's going on. If you think about research and the products of research, obviously it's the impact on patients' lives ultimately. That's often quite downstream of what we do. Another measure is, of course, publications on that research. And this is a graph which I've shown previously at some of the national venues with the OIF. And it represents one metric, the number of publications of papers over the 50-year history of the Osteogenesis Imperfecta Foundation. And under some of the arrows up on top, you see some of what I consider landmark findings, the cloning of the type 1 collagen gene mutations that cause the majority of OI, the application and report by Francis Gloria of bisphosphonates in treatment of OI. Roy Morello, our own group's work as part of a large collaboration, discovering some of the genes that lead to recessive OI and some of the many, now over 20 genes that contribute. And what you see is that there's been this steady growth, but there's been an inflection point in recent years. And what that's been driven by has been, in fact, support of the OIF, first with the linked clinical research centers. And then, of course, in 2014, the Brittle Bones Disorders Consortium. So I would say that certainly this is one product that you can see in terms of the momentum I talked about and the reports on scientific studies. So what have these studies been about? At the core of the Brittle Bone Disorders Consortium is what we call a natural history or longitudinal study. The goal is to follow over 1,000, close to 1,000 OI patients, and really to understand all elements of their health, but especially focus obviously on skeletal health, things like bone density, scoliosis, vertebral compression, and how this evolves in different types of OI and with different impact of different treatments like bisphosphonates. But it's not just about skeletal health. This core longitudinal study, which is participated by all the different sites. It includes things like hearing, lung function, how well we breathe, also clinical trials. I will probably touch on some of these later, but a clinical trial of different new drugs, for example, what we've tested, a drug called fresilumumab, which is something that blocks a growth factor called TGF-beta. And then most excitingly coming up, a clear aligner trial to try to fix malocclusion, something that really impacts quality of life in OI. There are also, in addition to these longitudinal studies, the clinical trials, also pilot studies, studies which are guided, frankly, by the reporting of OI families themselves. In the last study, work by Laura Tosi and others underscored this importance of pain and anxiety. And we now have support from a whole new NIH institute to begin to study anxiety and pain. There are pilot studies on heart and lung health, and this is an ongoing issue supported by the OIF. And so there's lots of these different unique and specific studies that are going on. Now, how do they differ from some of the work that's going on outside of the BBDC? So first, let me just say that there's lots of outstanding work going broadly in the community, and that's important for a healthy, scientific and advocacy community to have work going on throughout. One of the great values of this BBDC is numbers. We know that OI is a rare disease, and those of us who have been in the field for many years, we take care of a lot of patients, but that actually is a drop in the bucket when you think about the number of patients you need to truly generate hardcore evidence. I like to tell my families, and sometimes they get very stressed out, that I think a lot, but I know nothing. Well, that's actually the honest truth. And part of knowing more beyond nothing is, in fact, to do research on large numbers of individuals. And so it's really only by collaboration among all these sites that we can have large numbers. And therefore, we are able to perhaps confirm observations or conclusions from others, part of smaller studies, which is important because confirming and replication tells us evidence is firmer. It may be to modify what's been observed or concluded by other studies. And importantly, it is to test completely new ideas or new hypotheses, or point to new problems that were not appreciated before because of the numbers. I mean, a good example is when you give a COVID vaccine to millions and millions of individuals, you might see side effects that you would have never seen with 10,000 or 20,000 individuals. And while that may not change the risk benefit, but at least we know, and we may know to look for them and how to counsel our patients.
So, all right, so given the size of the OI population, how much more difficult is it to get the results you want, assuming we're looking at 50,000 people?
So, you know, that's a hard question. And the reason is it depends on what you're asking. It has to do with actually a major theme of what NIH wants us to do in the consortia, which is clinical trial readiness. A big part of clinical trials, as many have heard in the COVID era, is how do you test a drug or test an intervention and determine it's actually effective or efficacious? Well, parts of that is based on how a specific target population, in this case OI patients, respond or to a certain measurement. And what clinical trial readiness is exactly to answer the kinds of questions that you brought up. If we were to measure pain, or if we were to measure mobility, what's the variation we see in a large population of OI patients? And by knowing that, we can actually power, which is a very important word, which really means to design well a clinical trial that will actually get us to an answer. And this is why sometimes someone will say, well, we need 1,000 patients for the trial, or 500 patients, or maybe 20 patients. By having the kinds of natural history data we're talking about, we actually will be able to project or predict how we design the best trials to test different interventions.
Is that what makes these consortium studies so much more valuable than, say, a standard longitudinal study?
Well, again, I think that those are overlapping. Remember that the consortium study is a longitudinal study. So it's about, again, numbers. You know, when you have a consortium, which is a group of individuals, we'll be able to study 1,000 individuals closely over a long period of time, as opposed to perhaps a single site study, which may be more resource limited. I think the other elements about consortia is that you're bringing collective brain power together. You know, I myself may think, what's the best way to, get from point A to point B? But when you have first the 12 sites and all the investigators involved in the 12 sites, and then you bring the investigators outside of the BBDC into these meetings, you're really bringing collective brain power together to actually, ask what should be addressed in these longitudinal studies.
Looking at this graph, is when you reach that inflection point and you saw the spike in papers being delivered, is that a matter of more investigators, more researchers getting involved? Is it more population getting involved? Why did we see the spike?
I think it's, those of us or folks who are on the call in the financial world call return on investment. It's all of the above. I think the investment that OIF made, which is where the LCRC was really that first key investment by the OI Foundation and the Brittle Bone, the Children's Brittle Bone Foundation, that brought investigators together. As you generate more data, it's kind of like the idea of that if you build it, they will come. I think it's attracting new investigators. It's training new investigators. A big part of the BBDC is training the next generation. It's generating momentum for the families to, in fact, participate. While the longitudinal study focuses on 1,000 patients, the contact registry, which was born out of the first phase of the BBDC, which has now been sort of taken over the mantle by the OIF, has over 3,000 registrants, I think. And so again, it's a good example of where by this investment in this consortium and the research that's going on, we've been able to really gather momentum both inside, outside, all around the community. And that's what this, I think, graph represents.
And since he brought it up, and we haven't mentioned it in quite a while, if you have not joined the OI registry, please, 3,000 out of 50, we can absolutely use your help. So I'm sure Michael can post a link directly to it in the chat. So if you have not done it, as soon as we're off this call, please go do it. All right, back to the BBDC. Let's jump forward to sort of where we are now, now that we've gone past this inflection point. What impact do you think the BBDC, since its inception, has already had on the way OI is treated?
Sure. Well, so we can go to some of the papers that have been come out of the BBDC, and I think that excitingly, in the next series of conversations after me, I think my job is really to sort of give the overview. But you will hear from investigators from the BBDC who led different studies and talk about their published goods, sort of what your participation has led to. And there'll be a whole series of different conversations around the products of that work. So, for example, probably to the best of my knowledge, the most careful characterization of the dental features, the malocclusion, the craniofacial, meaning how the skull is shaped and functions in OI, has been reported in multiple papers led by Dr. Jean-Marc Retrouvé and his team. And so this is descriptive work, but again, in the vein of clinical trial readiness, it in fact informed this new exciting trial focused on aligners, clear aligners in trying to treat malocclusion and how to power that study. And so that's an immediate impact. And I know there's great excitement in the OI community in participating in this. The knowledge that will be gained will be able to be broadly disseminated to actually guide orthodontists to address something and to give them an approach using potentially these clear aligners, if this trial is successful, to address malocclusion, to really be able to chew and eat food. You can imagine how something we take for granted can have such a great impact on the life of an individual with severe OI. I think that's a really exciting example of how the longitudinal study and the different pilot studies in phase 1 has led to this real interventional trial that I think ultimately will have great impact. There's been impact on treatment. We just are about to submit the paper which describes the phase 1 clinical trial of this approach, this new approach of blocking TGF-beta in OI individuals to treat their bone disease and perhaps their pulmonary disease at some point. And we found that this drug, fresilumumab, is safe and well tolerated in OI adults, and in some of them have pretty dramatic increase in bone mass, even after a single dose. And so I think that's another example of actually bringing to the table a potential drug that could be further studied, and it would need to be further studied in what we call efficacy trials to potentially get at, the first FDA approval of a OI treatment. We have no FDA-approved OI treatments. It's important to keep that in mind. Bisphosphonates, which have been such an amazing advance, is not FDA approved. It's really a repurposing of an osteoporosis drug. There have been many others, and I don't have time to go through all of them, but quality of life measurements. And this was something started originally with Lortosian, looking at an important tool called PROMIS, but has extended to other tools. Again, different ways to measure what is quality of life in different aspects, and that can immediately be used in the context of clinical trials. There are growth curves that have been generated that help to determine and guide clinicians on whether a child with OI may be growing appropriately in the context of having different forms of OI. Continuing work on, I think, and here where we don't have definitive answers yet, but drawing attention to an issue that the OI Foundation themselves and with the families identified, which is the risk of cardiopulmonary disease. Well, that's a pilot that's ongoing right now. I think it's all about, evolving the idea of thinking about the disease, studying it in terms of in animal models and cells, but really converting that into knowledge about patients. And so I would say as a whole, there are so many issues related to different organ systems that affect OI that have now come out of the BBDC.
Giving you a magic wand, and based on the work that you've already done so far, the research and what you've already found out, where would you like to spend more time, more manpower, and frankly, more money?
Wow, that's amazing. Well, you know, if given unlimited resources, there's no question that the two, I would say, most important areas for us to tackle is to actually get an FDA approved and get many FDA approved therapies that improve bone health in OI. Because again, we don't have that yet. I think that, you know, what would be best is that there are many, many drugs available, and to pick the right one for the right patient. You know, this whole idea of precision medicine. We know OI is not one disease. OI is literally thousands of diseases. I think any of our families tell you that there's no two OI patients that's the same. And so in all the studies, what we're seeing, this heterogeneity, can we translate that into therapies, which in fact can target different types, you know, use drug A for this type or drug B for that type. I think that's the deliverable in medicine in general, but certainly I think for OI. I think the second is, of course, everything outside of bone. You know, OI is a heritable disorder of connective tissue, described beautifully by the father of genetics, Victor McCusick, in the 50s. And he recognized that this was not just about bone. And so I think focusing on all of these other things that impact the quality of life as OI patients live longer and longer, you know, as they become adults and they age, it's not just bone that's causing significant morbidity or impact on quality of life or these other issues. So I think, you know, focusing on this together with OI patients will be another very important goal for the future.
I hear this question a lot, and just out of curiosity, how important is it for you as a researcher or even a physician or an orthopedist for your patient to have genetic testing done to know their type?
So that's, you know, it's a very important question. As a geneticist, obviously, I think it's critical to have genetic testing. What I would say is that there's no issue that genetic testing gives you more information. And so if you believe information is power, then sort of a priori, having more information is a good thing. And that's perhaps more philosophical than practical. What I would say is there's great value for understanding, you know, clinical impact of that information. And if genetic mutation can tell us, well, we're more, you know, likely in OI type 1 versus in OI type 3 or in OI 3/4, or a form of OI that has more connective tissue features versus one that's more on bone. I would say that there's been now so many mutations described, especially in the collagens, specifically the collagen-related forms of OI, where that information can help us counsel patients. And I wouldn't say predict the future, because no one can predict the future, but give a picture of what the future may hold. So it's interesting that, you know, as a geneticist that studies many different genes and many diseases, albeit OI is closest to my heart, as Tracy Hart knows, OI and the collagen disorders is actually one of the best examples where genotype, the nature of the mutation, actually can tell you something about phenotype, the severity of the disease. So I think certainly there's that aspect of it. I think, though, the other component is this idea of, again, personalized care. If we can get there, there's no question knowing the mutation will be the first step in helping guide what type of treatment you have. For example, in the anti-TGF-beta trial that we're doing, because all of the mechanistic work we did in the laboratory showed that the most significant effect were in collagen and collagen modification disorders that cause abnormal overmodified collagen, we actually limited initially the inclusion criteria patients studied with those types of mutations. Now, you know, whether this treatment will work for other types will have to be determined. But certainly, even in our preclinical work, there's a suggestion that there will be specific mutations that will respond best with this type of treatment. And we know that even for clinical trials. If you may remember, one of the largest adult clinical trials that's been done was work that we did with Eric Orwell and Jay Shapiro and Sandra Schneidermani and others, when we studied the comparison of an FDA-approved osteoporosis drug, teriparatide, in OIs versus placebo. Very robust placebo-controlled trial. Folks have heard about that term, certainly in the COVID era. And surprisingly, we found that haploinsufficient type 1 OIs, individuals who have OI type 1, due to a decrease in the amount of collagen in their body, not the quality, actually responded very well. But the other types, type 3 and 4, the ones that we've targeted in the TGF-beta trial, actually didn't respond as well. So again, these are increasing examples coming out of research like what's being done in the BBDC as well as others, that genotype mutation can in fact begin to inform phenotype and potential therapeutic approaches.
All right, we have another question we've been getting quite a bit is what research is being done for the adult and or aging elderly OI population?
Well, so what I would say is a lot, certainly in the BBDC, because, you know, it's a longitudinal study and it's to study the life course of OI. It's not just in children. And so a large proportion of our subjects are, of course, adults, and it crosses the whole life spectrum. And currently, we are already following individuals up to as much as 7 years. We know that the grant is funded for 10, and so at least 10 if we're lucky, and we funded for another. We will follow as far as 15, and hopefully beyond via other mechanisms. So that is embedded in any longitudinal study. We are trying to study the effects of aging. You know, what is interesting, and this is something I've always sort of believed, and this is more, again, belief than science, is that conditions like OI are actually at some level a example or a model of accelerated aging, right? That's not too hard to believe, right? It's not such a big leap of faith. I mean, as we get older, we get fractures. And in OI, we get fractures really young and continue throughout life. And so I think that, you know, this idea that aging impacts OI, well, I think actually OI may be a form of accelerated aging. And I think as we learn about how to treat aging, it will impact our treatment of OI, albeit maybe even at an earlier time point. But it is the main focus of the longitudinal study. It's one of the major questions. How does, you know, aging impact all the different connective tissues? So, you know, make sure you participate in the registry. And if you're not in the longitudinal study, you know, reach out to a site and maybe we can enroll you.
We actually have a very specific question here. What are the recommended treatments for 65 and over age group with severe scoliosis and multiple compression fractures?
Okay, well, I don't want to get into specific recommendations. That's, you know, not generally a good idea because I haven't seen whoever asked that question, and there are a lot of things that could impact the answer. You know, it's certainly easy to say generically, there are drugs that you want to build bone health and bone mass. And so that's going to be part of it. There are going to be, you know, approaches to treat compression fractures where you can, in fact, inject, if there's significant collapse of that fracture of that vertebrae, you can inject materials to sort of strengthen it. There is no question that physical therapy, increasing core strength, is possible. Even in someone who's more mature, bracing could be considered. While bracing is something we think of in the pediatric age group, but to give support for the skeleton. So there are many, many possible interventions. There is no magic formula because you really need to talk with your healthcare provider, your specialist, who takes care of all of these issues to make a personalized plan, because you never know when sort of a generic intervention is contraindicated. So that's important to keep in mind.
All right, we have another question from Anne. She's interested in recessive inheritance. Is this new explanation for single cases previously thought to be the result of spontaneous mutation?
So absolutely. I mean, if one of the things I am proud of that our group contributed in that paper describing the recessive forms of OI, you know, it was thought for many years, not because it was bad research, it was excellent research, but again, you're limited by technology and the resolution power, that OI was a condition that actually had a high recurrence rate of apparently new mutations. So the idea that, you know, you have what we know to be mostly a dominant genetic condition, mutations of ectin collagen, but occurring recurrently in families who have parents who don't have any symptoms. And that was thought to be due to something called germline mosaicism. I'm not going to get into the specifics of that, but it was unusual that it was so high. Well, it turns out that many of those cases probably were more likely recessively inherited forms of OI. We now know that there are many recessively inherited forms of OI, meaning that in fact, both parents carry a copy of a mutation, not a collagen gene, but another type of gene, and that only when both copies are inherited, you can get that form of OI. And of course, because both parents are carriers, you can have recurrence in the family. And so, yes, absolutely, recessive inherited forms of OI probably explain the majority of cases where you have 2 or more children with OI born to apparently unaffected parents. Sorry about that genetic education piece, but.
Completely understand. All right, we have another question from Nicole. How does OI typically affect tendons and skin tissues that have lots of type 1 collagen?
So that's, again, an excellent condition. We just are in the process of publishing some papers on this, on especially the tendon work. So as you point out, type 1 collagen is in all over. It's not just bone. It's in tendons, ligaments, skin. And I think, again, patients and families with OI who live with this disease every day know that. And interestingly, when you think about tendon and ligament function in OI, sometimes it can be too loose. And in certain forms of OI, it actually can be a little too tight. And so it's not a simple answer that type 1 collagen weakens or strengthens it. What we do know is that collagen, type 1 collagen especially, in tendons and ligaments and in skin, can be modified differently. So they don't look exactly the same as in bone. And that's not shocking, because you think about what collagen does in bone, it's actually to be filled up with mineral crystals, right, to form hard tissue. It's not the case in tendon and ligaments, not the case in skin. And so we know that this whole idea that collagen has to be modified or changed occurs, and it's different in different tissues. And that can also lead to why there are different consequences of collagen mutations in these different tissues. Also, we now know in some of the work that we are just about to publish that just like in bone, when you have abnormal collagen in tendons and ligaments, you can actually affect signaling. So the TGF-beta work I pointed to was very much focused on what we did in bone. Well, it turns out not necessarily TGF-beta, but other mechanisms can be altered in tendons and ligaments, and I would guess also in skin. And so probably what this tells us is that when you change matrix, you not surprisingly change the communication of that matrix to all the cells in that tissue, and that then changes the function of the tissue. The good news is, by identifying this, we may have strategies to modify those signals and perhaps help with the tendon ligament problems, whether it's too loose or weak, or if it's too tight and contracted in different forms of OI. And similarly, while wound healing is not the major issue in OI, it is in connective tissue diseases in general, we know that there are patients who have some problems with it. And again, similarly, if we can identify the underlying mutation and it associates with a specific change in pathway in that tissue, we can develop therapies for it. And that's really sort of the whole translational research pathway that we all are trying to do here among the OI community.
Before I get to one more question, I do want to address one real quick. Carolina asked about, is there anything, the risk associated with pregnancy for a mother with OI? And just want to let you know, we do have another town hall coming up with Dr. Deb Krakow about women's health. We're probably not going to get into that tonight, but that is coming up. We will have a time. We will send that out. So definitely stay tuned for that.
I would just add that, and I'm looking forward to hearing that from Dr. Krakow, but that was a signal, a good example, because you asked about, well, how does that impact OI care immediately? That was a signal that Dr. Krakow identified as part of a pilot study that was done partly with the contact registry, and some of you in the community may have participated in it, which in fact identified an important element of pregnancy and risk of pregnancy. So, let's make sure you come to our talk on that and our conversation, because I think that's a beautiful example where I would say in general, the field has not been sensitive to some of the risks related to delivery beyond fracture. I'm not talking about fracture, but other issues. And I think her work in the BBDC really highlighted a potential important issue, which now is being studied in this phase.
Absolutely. Check your email. We will be sending that out fairly shortly. Are there any pharmaceuticals, drugs, over-the-counter drugs, or everyday products that someone with OI should avoid?
Well, I probably, that's a very, very broad question, because basically it's like, what's bad for me?
Right.
And there are lots of things that probably could be bad for oneself. Anything that certainly is bad for bone health, could adversely affect someone with OI. But I probably don't want to get into any specifics and give without evidence in hand.
All right. On the flip side, could you?
I would say don't smoke.
Yeah, fair enough. On the flip side, could you be more specific about drugs for building bone mass after age 50?
Sure. So, as many of you may know, there's two ways where we can affect how much bone is present. It doesn't affect the quality of the bone, but it affects how much bone we have. And bisphosphonates, a medication that many of you know and have used, and I think we all think has an enormous impact, especially in the children with OI, gives you more bone primarily, or maintains your bone, I should say, in adults, by blocking resorption. Those are the cells or mechanisms that take bone away. And it's a very potent anti-resorptive. One of the problems in adults, of course, as you know, is for all of us, whether you have OI or not, it is downhill after your 20s in terms of how much bone you have. And so because of that, even with bisphosphonates, it doesn't help you build significant amounts of bones. What it really does is slow down the decay. And not that we're all decaying, but it is an issue with the amount of bone you have. So of course, in the osteoporosis world, there's been a big push to study and develop therapies that build bone, which works on the other side. The osteoblasts, which makes bone, right? The guys that take away bone is osteoclasts, the cells that make bone osteoblasts. And in fact, and I alluded to this before, there are now FDA-approved drugs for osteoporosis that build bone. The best example that's been approved the longest is, in fact, teriparatide, which is an analog of PTH, parathyroid hormone. And in fact, when you inject this analog every day, it builds bone. And that was the trial we did several years ago, I alluded to. And so certainly, if you have OI type 1 and your doctor would like to build bone as opposed to block resorption of bone, I think teriparatide is a great option. Another therapy that has been approved by the FDA for osteoporosis is an antibody against sclerostin or anti-sclerostin. And this is, again, an anabolic therapy, and it's very effective, again, in building bone. As some of you may know, and maybe some of you are actually participated, there was and is a clinical development program by the company Mereo, and now Mereo together with Ultragenyx, to study anti-sclerostin. And so, again, I think the results from that early trial, again, it wasn't focused on efficacy per se, that we would think like a phase 3 trial, but it's very encouraging. And so, in the sense that, bone mass did increase in patients who were treated with the anti-sclerostin antibody. So I think, if that goes on further, that would be another approach. So I would say, in terms of bone building, those two are examples. Our anti-TGF-beta also actually had an anabolic effect, building more bone in adults. So the great goal should be that we have many of these therapies, and to determine ultimately in downstream studies which work best in which forms of OI.
I have another question for you. Are there any concrete genetic treatment plans on the horizon?
So gene therapy is a very hot area. And in fact, many of us have already had gene therapy, right? What is messenger RNA vaccination but a form of gene therapy? So we actually all have had gene therapy, or unless if you've had the Pfizer or Moderna vaccine. I think that The other type, so you could get a nucleic acid or some way of delivering a nucleic acid. And I would say that there have been gene therapy studies on that front. It's a tough bar to try to, make more type 1 collagen. And I've always said this since I do study gene therapy, because you have to target so many cells in the body, right? Unlike doing gene therapy for blindness, where you just target the cells in the back of the eye. Or if you're doing gene therapy for liver disease, where you just need to target the liver, we have to target cells in the whole skeleton. And on top of that, those cells are embedded in a pretty tough matrix. It's kind of like a castle and a moat you have to get through. So I would say that gene therapy as an approach is not easy for a disease like OI. Now, there is a flip side, and there's cell therapy. It's not gene therapy, but you're giving genes in cells. And many years ago, some of you may know, there was an attempt to do cell therapy using sort of stem cells via bone marrow transplantation type approaches. And we now know in Europe, there have been there's been cell therapy trials in utero. What I would say is it's unclear how effective those will be. It's really hard. Also, again, you've got to get into the skeleton and get enough of the skeleton repopulated. So I would say that, certainly in the near future, the hope is probably greater for drug therapy as opposed to genetic therapies, but I would never rule it out. And part of that is because of the great science that's supported in our country and throughout the world. Who would have thought that, millions of people would get injections of messenger RNA? And so I would say that it doesn't take a lot. It takes one great discovery, and it could certainly change our approach to things.
We have another question asking about collagen folding and the fundamental defect.
Well, that's a great question. As many of you know, when we have severe OI caused by collagen or collagen-related genes, the severe forms, OIs 3 or 4, are due to changes in the collagen that affect its folding. And now we actually see other genes in that affect collagen folding and transport. So it's a whole class of disorders. I think that there are many consequences of when you have collagen that's not folding correctly. First, you have bad collagen in your bones, and I think that affects quality. And so we, up to now, have no real way to treat and correct, quote, the bad collagen so that we can directly impact quality of bone. So that's, I would say, another one of those important targets scientifically. What we do know is that when you have this collagen that's not folded right, it has many effects downstream. It can affect the stress state of a cell, of the bone cell. Cells have to kind of clear garbage away. Not that collagen is garbage, but think of it where you've got to normally clear things from a system. We now know that when you have proteins that are not folded right, you can stimulate stress in the cell. And there's work by excellent investigators who are trying to understand that stress and see if we can treat that stress to improve bone. So that's certainly one very ripe area of unfolded collagen and how that impacts potential science or function. Another area of how that unfolded collagen can affect the bone is our own work, which I've alluded to, about how it changes signaling. If the collagen is not folded right and it's structurally abnormal in your tissues, it affects how it binds to growth factors. And this is the basis of this anti-TGF-beta treatment. So that's certainly another consequence now outside of the cell. And probably the final consequence of this idea that collagen is not folded right is its function as a scaffold for minerals. Remember that in bone, you have to deposit this mineral onto the collagen scaffold. And so again, when you have this misfolding causing altered modifications in structure, it's kind of like having the girders of a building being structurally abnormal. Well, the things that you put on that girder will be weak. And so again, that may lead to this problem with mineralization that we see. And so it is at the root, I think, of many of the issues and problems we see in OI. And if we could fix that, I think whoever asked that question has a great idea of trying to fix many of the downstream pathomechanisms, as we call it, mechanisms that lead to the pathology that we see in OI.
All right, I'm going to go back to one of your earlier answers from Sarah Reed. Hi, Sarah. What is the timing of the anti-sclerostin drug becoming widely available for treatment, assuming that the drug trial continues to be successful?
So let me answer that practically, and then theoretically. Theoretically, because I can't predict the future, is in any drug development, after you have basic safety studies, which is what's been done. I think the anti-sclerostin studies have been done to date, at least what's been reported publicly, suggests it's generally safe, which is not shocking because again, it's FDA approved in a large population for osteoporosis. So there are cardiovascular signals in the large studies, but again, if you study lots of people, you will see some rare side effects. So I do think that there is certainly enough scientific evidence to move forward into what we call phase 2 and phase 3 trials. And I think these are the ones that are being contemplated. And so those trials, of course, test the right dose that should be used and the efficacy. And that occurs, generally in this idea of a controlled, placebo-controlled study, meaning there are individuals who get the drug and individuals who get some placebo. And if that shows it works, then that's when, given appropriate efficacy endpoints, then it can go forward for potential FDA approval. Now, this is where the hang-up is. What is the right efficacy endpoint? Because again, if you remember, I talked about one of the really important elements of the BBDC is clinical trial readiness. Well, bone density, the measure we use every day in all of our patients to help guide therapy, is not accepted as a primary endpoint right now by the FDA to approve a drug like this. And so what that leads us with is, well, then what is accepted? Well, in most bone health related drugs like osteoporosis, the major endpoint is fracture frequency. And so, do you decrease the number of fractures with your drug? One of the challenges of studying that endpoint in OI patients is, of course, fractures can occur hundreds of times in someone with OI type 3. It may occur once or twice a year, if at all, in someone with OI type 1, in an adult, let's say. And so because of that very skewed distribution in different patients, it's very hard to power a clinical trial. And if you need 10,000 OI patients to participate in a trial, you can imagine how likely that will be done. And so there are a lot of variables that I think will impact whether a clinical phase 2 or 3 trial can be designed and how it's designed. And we hope that the data we've generated in the BBDC may help inform that. Typically, it takes oftentimes, in the range of 3, 4 years or 5 to get trials like this done and ultimately approved by the FDA. So again, that's not an average. It's just think about the range in the kinds of diseases we're talking about. It could be 10, but I would say it's in the several year range if in fact it gets done. What I would also say though is anti-sclerostin, just like teriparatide, are FDA approved already. So in theory, and I'm not advocating for this, but in theory, this is the practical answer, and osteoporosis are all a doctor, and I have considered in my own patients, could choose to elect to give this off-label. The risk, of course, is that you don't know whether there could be some very significant reactions that occur. That's unique to OI that we are not aware of because there have been not trials completed. There's always risk doing a drug treatment off-label, meaning it was not studied and fully vetted by the FDA for that indication. But because they are FDA-approved drugs, it can be prescribed by a physician off-label. And in fact, bisphosphonates have been prescribed off-label for OI for decades, because it's not FDA approved. So I would say that, those are sort of where we are right now with some of these newer anabolic agents. But we're very hopeful that all of them will move forward to phase 2 and 3 trials. And I think it's in all of our interest to collaborate to try to do that, because the endpoint is to have as many therapies as possible.
All right, jumping over to hearing, and specifically hearing loss in adults. Is there anything that can be done to slow it or prevent it? And what type of effect might drugs like bisphosphonates have on it, given that it may affect the bones in the ears?
Yeah, I don't want to speculate. That's not my area of expertise, but I can tell you that that's something that we are following and looking at in the BBDC, because so many patients have had bisphosphonates, and we are monitoring hearing over the longitudinal study. There is no question that autosclerosis, or where the hearing, the bones in the ear that mediate hearing are affected and contribute to the loss of hearing. Whether there are drugs to slow that down, I think, are important questions. What is the impact of bisphosphonates longer term in patients with that? We don't know the answer to that definitively, certainly. And so I think these are some of the things that we want to begin to address in BBDC. Right now in the BBDC with things like hearing, we've primarily described what we are talking about as cross-sectional data, meaning that we've looked at a large population and can describe things like severity, incidence, complications, and it's a good baseline. The longitudinal part is the big deliverable. This is what the exciting part will be in the years to come. That's following individual patients over many years and seeing how does their hearing evolve. And again, those are some of the data that I think will be very helpful in potentially answering questions like yours. Because, for example, if we look at individuals who were on bisphosphonates versus that weren't, and we follow their hearing over time, if we see differences, one could then begin to make a hypothesis or a correlation, not causality, that bisphosphonates could impact that. I think there's certainly been discussions on that front, but I personally don't know. There may be others who perhaps have an opinion on this, but again, I would say that it's only with large numbers do we kind of evolve from this idea of thinking to knowing.
Right. Another audience question. Taking the teriparatide, after 2 years, it did increase my bone density. How can I maintain that bone density even with taking zoledronate every year?
So as the osteoporosis world has taught us, with almost all the anabolic therapies, one usually has to follow with consolidation. When you build bone with an anabolic therapy in an individual with generic osteoporosis, you will invariably stimulate after that treatment period, which can be 12 or 18 months or 2 years, and generally it's in the 12 to 18 month range. You will stimulate bone resorption. And the reason for this is a physiological principle we know well in bone biology, and it's called coupling, that bone forming cells and bone resorbing cells talk to each other. And so when you rev up those bone forming cells with teriparatide or anti-sclerostin, after a while, they will eventually rev up bone resorbing cells. Probably teriparatide does it more effectively, communicate and rev things up a little bit faster, but it does happen. And this is why, not surprisingly, you are on zoledronic acid, anti-resorptive, to consolidate or maintain that. I think the bottom line is that's the standard. Now, how long you need to stay on it? Can you go back on an anabolic? These are all questions that I think are, unknown really, and are driven by empirical, I would say, experience really. I guess all experience is empirical, but it's one of those questions. Again, hopefully we'll begin to collect some of the data in the BBDC. Certainly in the osteoporosis world, on lots of patients is being addressed, and they have been sort of approaching this idea of, anabolic followed by consolidation. Do you then follow with another anabolic? But the best we can do is extrapolate from experiences in the osteoporosis world, which, may be okay, but again, may not. So again, underscoring the importance of why be involved in longitudinal studies like the BBDC.
All right, another audience question. This is probably the last one we have time for. OI type 6 may be caused by mutations in the, and I hope I pronounce this correctly, SERPINF1 gene. Could you please talk a little bit about this gene?
Sure. So OI type 6, specifically a form of OI that Francis Gloria, Frank Rausch, and others described well, is caused by mutations. It's a recessive form of OI. It's caused by loss of function of this protein SERPINF1, or another term for it more well known is pigment epithelial derived factor, PEGDF, pigment epithelial derived factor, PEDF. Sorry, SERPINF1. It is a protein that floats around in our blood, and for many years we didn't know anything, that had anything to do with bone. It was actually studied in nerve cells. It was studied in the eye, given the name pigment derived because it's expressed in the back of the eye. And it's a very interesting protein because it's one of the strongest antagonists, actually, to another secreted protein called VEGF. Bottom line is that OI type 6 patients are missing this protein, and because they're missing this protein, their bones don't mineralize the same way. And I think that the interesting point about OI type 6 is a question of whether replacing it could fix OI type 6. I think conceptually the answer is yes, because if you're missing a protein in the blood, putting it back should make it better. So I think that that's certainly a reasonable future direction. Now, we've done experiments which at least showed that it wasn't as simple as that. We expressed the protein from the liver, and that did not fix the issue in the bone. Having said that, that's not, I don't think, the definitive answer. Other studies published by others suggest that if you express it at a different level, maybe it does. So what I would say is, at this point, the preclinical evidence is unclear, which is science. It's completely okay. This is why we have to do it. I would say that the question of whether replacing and putting PEDF in the blood alone would fix OI type 6 is still unknown. It may be that it's not simply that. It may be you might need to put the right amount or the right form, or it may need to be made in the bone, for example, because we know that a lot of these factors that circulate in the blood may not be active enough in the bone, and there could be what we call compartmentalization. So there are a lot of different things I think that would need to be studied, but they're ripe for study. So it's an exciting area.
I think we are just about out of time. Dr. Lee, obviously cannot thank you enough, and this is why we love having you on for hours and hours and hours. Next up, we are going to be doing this on a regular basis. We also have national conference coming up. So for those of you, check for more information. We have women's health, as we mentioned, coming up with Dr. Dip Krakow. We have dental coming up with Jean-Marc Retrouvé. We have other ones coming up. We will be doing this as frequently as we can. When we do post them, you can ask questions ahead of time on BoneLink at oif.org. You can submit them here on the chat. We have folks from Japan, Shiro. It's not even 8 o'clock in the morning yet. We have folks from Indonesia. It's not even 6 a.m. there yet. This is global. Everybody can join. So this is not just an American conversation. So anybody from anywhere, tell all your friends. We're more than happy to have you. So thank you, Tracy. Thank you, Michael Stewart. It has been amazing to be able to spend this much time with Dr. Lee. And we really want to thank you. Michael, I'm going to turn it back over to you.
Great, thank you so much, Cameron. So just for everyone on the call, thank you so much for joining us. We have recorded today's session. We're going to post it in the next couple of days onto the OI Foundation's YouTube page. Also, again, as Cameron said, if you have any questions, email us at bonelink@oif.org, and we want to connect with you. If you want to find any of the OI Foundation's resources or look at more information on the BBDC, you can go to the OIF's website at oif.org, and we have lots of things related to COVID-19 and beyond, and even in the larger context of all the research Dr. Lee was talking about. So for those of you still with us, thank you so much. Have a great evening. Stay safe, wash your hands, wear 2 masks, and get vaccinated. And hope to see you all soon. Thank you.
Happy Wishbone Day. A little late on that one.
Yeah, happy Wishbone Day, of course.
Go Yankees.
Oh, come on. So not fair.
On that note, good night, everyone.
Good night, everybody.